Hi all of you. In this video, we're going to learn about epigenetics. So what is epigenetics? Epi means above, right? So it is above genetics, which means that it deals with the study of alterations. Yes, study of heritable changes or heritable alterations that happen around the gene sequences and not involving the nucleotides of our gene sequences, right? So we all know that if there is any alteration in the nucleotides of the gene sequences, that will be heritable, right? But this epigenetics deals with the study of any changes that are heritable, but they are not involving the nucleotide sequences, right? So if without involving the nucleotide sequences, it is changing the genetic expression, right? Whether the a particular gene can be expressed or not. That is what is given by the heritable changes, and those changes are not involving the nucleotides of the gene sequences. So let's again define this epigenetics. These are heritable changes in the DNA, right? That alter the gene expression. Yes, but without alteration in the nucleotide sequences. Yes, without alteration in the nucleotide sequences. So they tell us about whether a particular gene can be expressed or not, whether it can be transcribed or not. Right. So based on that itself, it is heritable. So suppose in a particular generation, the chromatin is very condensed, it is transcriptionally inactive. So as the inactive form itself, it is given off to the daughter cells. Okay, so as an inactive form itself, it is given off to the daughter cells. In that way, a transcriptionally inactive form is being carried to the daughter cell. Yes, it is transferred to the daughter cell, but there is no involvement in the involvement or mutation in the nucleotide sequences. There is no involvement in that. It is only the transcriptionally active chromatin is changed, but without involving the nucleotide sequences. How are you going to transfer this information? How are you going to transfer this sort of gene expression? Right? How are you going to do that? By modifying the chromatin, right? By modifying the chromatin. So there are three main factors that are involved in epigenetics. So only these three main factors, of which the DNA methylation and chromatin remodeling are very important. And the third one will form a minor role, right? So the first one is DNA methylation. So what do I mean by DNA methylation? So along the promoter regions of your transcriptional unit, along your gene, yes, there will be CG islands, right? There will be CG islands. So our books can give us like CPG, right? What is CPG? They are CG islands with just to differentiate between, that is a phosphate link between the C and G, they are giving us as CPG islands, right? So along the CPG islands, when the cytosine, yes, when the cytosine is methylated, when the cytosine of the DNA along the CPG island is methylated, that is what is there, that is what is being called as DNA methylation. So that DNA methylation can help in the alteration of gene expression. Yes, so this hypermethylation of this DNA can involve in gene silencing, right? Hypermethylation can involve in gene silencing. All right, so what is yes, let us come to the chromatin remodeling in the next slide. But DNA methylation, that is, since because it is methylation, it is catalyzed by methyl transferase enzyme. Yes, so there is a methylation of cytosine that forms the 5-methyl cytosine in the CPG islands that plays a role in the gene expression. So it particularly inactivates transcription. All right. The second point is chromatin remodeling or post-transcription translational modifications of histones that are helping in the regulation of gene expression, right? So this is also one main factor, the second main factor that is involved in epigenetics. So what is this chromatin remodeling? Yes, what is chromatin? In the previous session we had seen that chromatin is of two types. Is it not? Chromatin is of two types. What are the two types? One is the euchromatin structure and the other one is the heterochromatin configuration, right? What is euchromatin? What is heterochromatin, all of you? Euchromatin is nothing but the decondensed form, right? The decondensed form of the chromatin, and heterochromatin is the highly condensed or tightly packed form, right? So decondensed form of chromatin is transcriptionally active. Why is it transcriptionally active? Because it is decondensed. It will allow its areas to be exposed to the transcriptional factors, right? So that is transcriptionally active. And heterochromatin conformation is tightly packed. It is very condensed and it does not expose the DNA to be acted upon by transcription factors. So it is transcriptionally inactive, right? Transcriptionally inactive. So what is chromatin remodeling? That is the conversion between this euchromatin of heterochromatin vice versa is what is the chromatin remodeling actually, right? So that will there are transcription associated structural changes that happen in the chromatin. That is what is chromatin remodeling. So in the simpler terms, it is the conversion of euchromatin to heterochromatin and vice versa. And what are the factors? Three main factors that help in chromatin remodeling. One is the position of nucleosome, right? What is a nucleosome, all of you? What is a nucleosome? Nucleosome is made of, yes, the long stretch of DNA. The long stretch of DNA, yes, it undergoes the first level of compaction in which the histones, right? The histone proteins form an octamer, right? Histone proteins form an octamer and the DNA is wound around this. Yes, and the DNA is wound around this, around the histone octamer. This structure is what is called as the nucleosome, right? So the positioning of the nucleosome. So if you're going to alter the positioning of histones, how these histones are how histones are tightly bound to this DNA. We all know histones are very positively charged because it is rich in basic amino acids like your lysine and arginine, and DNA is negatively charged because these two charges are opposite. They tend to bind tightly, right? So if you're going to remove the charges, positive charges of histone, then what will happen? Yes, if you're going to reduce the positive charges of histone, then what will happen? The DNA and the histones will be loosely bound, right? So that will bring about decondensed form, right? That will become transcription, that a particular gene will become transcriptionally active, is it not? So the positioning of nucleosome will alter the chromatin conformation. Also, presence of histone variants. Yes, and covalent modifications of histonucleosomes. So all these three factors help in chromatin remodeling. All right. Yes. So let's see a few of the histone modifications. So what is the important thing that you should know of histone modifications? So there will be a question called histone code. Yes, there will be a question called histone code. What is that histone code? So all that I'm going to describe now on this histone modification will be the histone code, right? So histone code is nothing but that histone code explains us about the involvement of this histone modifications, the covalent modifications of histones, involvement of these in the epigenetics. That is what is histone code, right? So histone code along with the DNA methylation, and the third point, what did I tell you? It is about the involvement of microRNA. Is it not microRNA or the long non-coding RNA involvement of this? So along with these two are going to give the epigenetic modifications, right? So of which histone code is very important, and histone code tells us about the histone modifications. So what are the different histone modifications that can happen? One is the acetylation, right? What is it? Yes, what is it? Let us list out. Histone, the first modification is acetylation, and the second one will be methylation. Yes, methylation. And third one will be phosphorylation. Phosphorylation. And the fourth one will be histone acetylation, methylation, phosphorylation, ubiquitination. Yes, and then it will be somylation and ADP ribosylation. Yes, so these are different histone modifications. So they bring about changes in your histone. So what are the different histones that we have? H1, H2, A, 2B, 3 and 4, is it not? So if you bring about changes in these histones are important in the formation of nucleosome, and nucleosome is important in the formation of or the packing of DNA in the form of chromatin. So if you're going to modify the histones, that is definitely going to modify the chromatin, right? This is going to bring about chromatin remodeling. So that is what we want. Now, so histone modifications. Histone acetylation. What is the first one? Histone acetylation. So I had already told you the histones are rich in lysine, arginine. Yes, the basic amino acids like lysine and arginine. So if you're going to attack this lysine, if you're going to acetylate this lysine at the amino terminal of your H3H4, like most commonly acetylation of this H3H4 that takes place of the lysine residues. Mostly these basic amino acids are found in the amino terminal end of your histones. Okay, amino terminal end of your histones are rich in basic amino acids, right? And that is catalyzed by histone acetyl transferase. Yes, how is it abbreviated? HAT, histone acetyl transferase. So what happens? Acetylation is CH3COO minus, is it not? So that decreases the positive charge of your histone and that will relax the binding with DNA. So that stabilizes what structure? It is euchromatin structure. So if the euchromatin is what? It is the decondensed form of your DNA. So the transcriptional factors can come and act on the DNA. So this euchromatin structure is said to be transcriptionally active. Okay, so histone acetylation brings about euchromatin structure conformation and that is transcriptionally active. So you can even reverse this by the help of the enzymes histone deacetylase. What is that enzyme? Histone deacetylase. All right, that will stabilize your heterochromatin structure. The second one, what is the second one? It is going to be histone methylation. So histone methylation can actually favor your euchromatin formation or your heterochromatin formation depending on which residue or which histone is methylated. Okay, so that is catalyzed by your enzymes methyltransferase. So histone methylation can involve lysine or arginine. So if you're going to methylate lysine 4, okay, that is going to favor acetylation. That will recruit the histone acetyl transferase and that is going to favor histone acetylation. So if it favors acetylation, then what structure is favored? Euchromatin conformation is favored, right? And that becomes transcriptionally active. Okay, so the example of which is H3K4 and H3K36, which means that histone 3, lysine 4 of histone 3 or lysine 36 of histone 3, if that gets methylated, then the euchromatin structure is formed. Now, if you're going to methylate lysine 9, okay, that will favor heterochromatin structure. So that becomes a gene silencer. Alright, that is going to become a gene silencer. Heterochromatin structure, transcriptionally inactive form. So not just lysine 9, can be even lysine 27 of histone 3 or lysine 9 of histone 3, right? So these are some of the examples which helps us understand that the methylation can favor euchromatin or heterochromatin depending on which residue is methylated. Depending on which residue is methylated. Alright, it is again catalyzed by your enzyme methyltransferase, right? Yes. The next histone modification is going to be phosphorylation. Phosphorylation of serine and threonine. Yes, phosphorylation, what is the enzyme? It is kinase. And dephosphorylation, what is the enzyme? It is phosphatase. Again, this modification is going to be reversible. So the main role it has in DNA repair, but also in the gene transcription, also in the regulation of gene expression. Okay, so it depends, it also involves, like similar to methylation, transcriptional activation or repression depending on which histone is phosphorylated. right? So that is phosphorylation of serine or threonine residues. The next one is ubiquitination. Ubiquitination means ubiquitination of lysine. Okay, addition of this ubiquitin protein to the lysine residues that allows access for enzymes for transcription. So it is going to stabilize the euchromatin formation, right? Euchromatin formation. Next is summoylation. Summoylation, what is sumo? SUMO, small ubiquitin like modifier, that is what is summoylation, and that will cause transcription repression by recruiting deacetylases. Okay, if you're going to recruit histone deacetylase, that is going to decrease the acetylation, so it becomes a transcriptionally inactive form. Yes, it is going to favor heterochromatin conformation, right? The next one is ADP ribosylation. The H1 histone 1, if you're going to ADP ribosylate, then that is going to cause decondensation of chromatin. All right. So these are the different histone modifications. So you have so importantly you have to understand on. If you can look into this picture, this is the first picture tells us the condensed form. Yes, condensed form, it is the heterochromatin conformation. Yes, heterochromatin. The second one, this picture tells us the euchromatin, it is the decondensed form. Okay, so if you can phosphorylate or acetylate or methylate. What is this methylation? Methylation at lysine 4. Okay, so these things can cause can favor euchromatin structure, whereas heterochromatin structure, methylation at lysine 9, that is going to cause heterochromatin structure. So even about DNA methylation, what happens in DNA methylation? DNA methylation is going to favor the heterochromatin structure only, right? DNA methylation of your CPG islands, cytosine residues of your CPG islands is going to cause favor heterochromatin conformation. All right. Yes. So